Welcome to video tutorial of NIG Academy. In this video lecture, we will talk about structure and function of nucleic acid. You know, in a life science, particularly in molecular biology, there are different molecules of life, such as carbohydrate, proteins, lipids, and nucleic acid. Before I provide a detail about nucleic acid, I want to tell you something about molecular biology. You know, molecular biology is a science that study about the molecules of life. Especially, it focuses on molecular understanding of process of replication, transcription, and translation. So it focuses especially in nucleic acid, the DNA and the RNA. So before I proceed to the detail of the lesson, I would like to remind you to subscribe, share it to your friend, and comment on this video. I hope this video lecture definitely broaden the knowledge of understanding of biology or biotechnology student. So let me proceed the lecture. As I've told you, this nucleic acid is one of macromolecules in molecular biology. Actually, it was first identified or discovered by scientists we call Michery in 1869. What he did was he isolated the nuclein from the nuclei of pus cell. So there are different chemical compounds. Among that, there is a phosphorus. He said a phosphorus containing substance is present in the cell that is responsible for the process of inheritance. Actually, at that time, he had no understanding about the DNA, but he was able to isolate what we call a nuclein from the nuclei of pus cell. So there is something which is responsible for the process of inheritance. So the nuclein was shown to have acidic properties. Hence, it became called nucleic acid. So what is a nucleic acid? Nucleic acids are macromolecules that store and transmit genetic information. The genetic information. I will come later on what we mean by genetic information. So anyway, it is a macromolecules. It stores something that is a genetic information. Then it will transmit that information from one generation to the next generation. So anyway, this compound is a polynucleotide. It is very huge macromolecules. Its building block is nucleotide. So a given nucleotide is composed of nitrogenous bases such as adenine, guanine, cytosine, thymine, and uracil. In addition to this, there is a sugar molecule, especially the pentose. There are ribose and deoxyribose. This sugar is again one component of nucleic acid. The third one is phosphate group. So a given nucleotide is composed of these three things: four of nitrogenous bases, pentose sugar, and phosphate group. In generally, there are two types of nucleic acids: deoxyribonucleic acid, ribonucleic acid. So, which is abbreviated as DNA and RNA. Let us see what we mean by DNA. DNA, it is deoxyribonucleic acid. It is a genetic material in any eukaryotic cell. When I say genetic material, it means that it is a material that can store certain information and it can transmit that information from one generation to the next generation. Because when we see the DNA, in the nucleus there is a chromosome. On the chromosome there is histone and the DNA, especially in eukaryotic cell. So this DNA do have what we call a gene, a millions of gene. So gene is a segment or a portion of DNA which store and transmit hereditary information. So DNA contained different millions of gene. So it is that thing which transmit hereditary information. Because of that reason, DNA is a genetic material in any eukaryotic cell. Actually, in some viruses, DNA may not serve as a genetic material. Rather, RNA serves as a genetic material. There is so many evidences for this. From that evidences, I'll focus on the evidence by Griffiths in 1928. This scientist carried out an experiment on mice by using Streptococcus pneumonia. What he did was he took this bacteria strain and he cultured in his laboratory. After that, he tried to infect the mice. He tried to infect his mice. His intention was to identify this hereditary material, how information is stored and transmitted. So he carried out these things in his laboratory. So virulent strain he designed as S, non-virulent strain as R, S smooth colony, R rough colony. So in his experiment, he cultured Streptococcus pneumonia, non-virulent rough strain, and he infected it into mice, but the mice survived. Again, using the same procedure, he cultured virulent strain, which is smooth. Then he tried to infect mice, but the mice dies. Again, he used heat-killed virulent strain. He infected mice, but the mice survived. At the end, he took this heat-killed virulent strain and non-virulent, which is a live rough strain, and he mixed it together. Then he infected a mice, but the mice dies. Mind you, in a virulent strain, mice dies. In non-virulent strain and heat-killed virulent strain, mice also dies. And this, dead cells from a donor virulent strain of bacteria were implanted into a recipient non-virulent strain to induce what you call virulency. There is something here I want to recall you. That is what we call a transformation. Something is transformed from virulent strain, from the virulent strain into non-virulent strain. Because in non-virulent strain, mice survived using virulent strain, but mice passed away. So there is something which is transformed from heat-killed virulent strain to non-virulent strain. Because of that reason, the mice died. So that is what we call a transformation. Something is transformed. There is some structure which is responsible for the process of transformation. In 1953, James Watson and Francis Crick reduced the structure of DNA using evidence from few scientists, such as Irwin Chargaff and others. When you see Irwin Chargaff also carried out certain experiment, and he finally concluded that the amount of adenine is equal to amount of thiamine. The amount of cytosine is equal to amount of guanine. Mind you, I will come later on. You know, adenine pair with thiamine, cytosine pair with guanine. Actually, he had no understanding about this pairing process, but he gave a clue, a hint. So he determined that the amount of adenine is equal to amount of thiamine, the amount of cytosine is equal to amount of guanine. But the amount of adenine plus thiamine is not necessarily equal to amount of guanine and cytosine. That rule is what you call charge of rule. Charge of rule. So you know, nowadays guanine pair with cytosine, adenine pair with thiamine. The other scientists, Franklin and Wilkins, also carried out an experiment. They performed X-ray diffraction studies. Then they identified three-dimensional structure of DNA. Three-dimensional structure of the DNA. So they try to reveal the image of DNA. What DNA looks like. What DNA looks like. The helical structure. Three-dimensional structure of the DNA. Three-dimensional structure of the DNA. So they discovered that DNA is something helical. It is twisted around each other. It is helical. It is helical. So they discovered that the molecule do have diameter of 2 nanometer and makes a complete turnover at every 3.4 nanometer. So from these evidences and from other evidences, Watson and Crick proposed double helical structure of DNA. So now DNA is a chemical form of molecules that store and transmit genetic information, which is used in the process of development, in the process of development and reproduction. You know, in that process, I will provide the next video. There is a process of what we call a replication. There is a process of transcription. There is process of what we call translation, which is the process of protein synthesis. So in such a process, especially in a replication process, so DNA store and transmit hereditary information. There is what we call a cell division. So I may provide that video another time. So anyway, DNA is a genetic material. It is store information and it transmits that information. So it is very important in the process of development, function and reproduction. So since it is one type of nucleic acid, its building block is nucleotide. So what is contained in DNA structure? One nitrogenous base, pentose sugar and phosphate group. Pentose sugar and phosphate group. This is a nucleotide. This is nucleotide, mind you. Nitrogenous bases, pentose sugar. In this case, it is a deoxyribose, and the phosphate. Here, its name is deoxyribose because it means hydroxyl group at the carbon number 2. This is carbon number 1, carbon number 2, number 3, number 4, and number 5. So at the carbon number 2, there is no O or hydroxyl group, OH. So that it is a deoxyribose. Its name is from this deoxyribonucleic acid. Deoxyribose. So this is a nucleotide. So when you see this detail of nitrogenous bases, two of them are what you call a purines, adenine and guanine. Because in this purines, there is what you call a double ring structure, which is a dicyclic. It has 4 nitrogen atoms at the position of 1, position of 3, position of 7, and the position of 9. So there are nitrogen atoms. This is the feature that make it differ from other. Again, when you say the attachment of this nitrogenous base to the sugar, just it attached to the sugar at the position of first carbon of pentose sugar. That is um, this one. This is the first position of pentose sugar. So attachment takes place there. And ninth position of purines. Ninth position of purines is this around. So attachment is carried out. So anyway, adenine and guanines are what you call a purine. The rest, cytosine, thymine, and uracil. Uracil, particularly, it is only found in RNA. So they are what you call a pyrimidines. They are not double ring. They are a single ring structure. They are monocyclic. Unlike that, they are monocyclic. It has 2 nitrogen atoms at the carbon number 2, number 1, the carbon number 1, and at the carbon number 3. At the carbon number 3, there is 2 nitrogen atoms. So the attachment is at the first carbon of pentose sugar and first position of pyrimidine. They are what we call pyrimidines. The sugar molecules in DNA it is a deoxyribose, because as I have told you, the at the carbon number 2 there is no hydroxyl group. Because of that reason, it is deoxyribose. Unlike RNA is ribose. RNA is a ribose. Okay. The last one is a phosphate group. It is PO4. It is a negative charge. Actually, I come later on this negative charge of phosphate. So here, as you can see from this, this is pentose sugar. Here, nitrogenous bases. It could be adenine, it could be thymine, it could be cytosine and guanine, and so on. So it attached at this position, carbon number 1, number 2, number 3, number 4, and 5. At the carbon number 5, the attachment of phosphate could be carried out. Here, it is a deoxy. Here, at the carbon number 3, there is free hydroxyl group. The new phosphate will be attached at here. Mind you, this is one nucleotide. This is another nucleotide. This is also another nucleotide. So this is what we call a polynucleotide. So new nucleotide will come and attach at these 3 positions. Because at this 3 position there is free hydroxyl group. There is a free hydroxyl group. So new phosphate will be attached at this position. Then it forms nucleotide. Another phosphate will be attached in such a way it forms what we call a polynucleotide. Actually, this DNA it is a double ring structure. This it contained what we call sugar phosphate backbone. It is a double ring. There is a sugar phosphate backbone. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. This side also sugar, phosphate, sugar, phosphate, sugar, phosphate, and sugar. So it is a double ring. Internally, there is nitrogenous bases. Those are oriented and inward. They are connected together by hydrogen bond. Mind you, charge of site, adenine equal with thymine, guanine equal with cytosine. Nowadays, as you know, thymine should pair with adenine because there is a double bond here, so their bond match with one another. They are connected by hydrogen bond. Here is a double bond. Cytosine and the guanine, there is a triple bond is here. Anyway, nitrogen bond connect these 2 strands together. Not only this, there are another bonds. That is what you call a glycosidic bond. Glycosidic bond, it is a bond that connect sugar molecule to nitrogenous bases around this area. They connect nitrogenous base with the sugar molecules. That is what you call glycosidic bond. And also, there is a bond that connect phosphate to the sugar molecules in both sides. Because of that reason, we call it phosphodiester bond. Phosphodiester bond. Here, carbon number 1, number 2, number 3, number 4, and number 5. So at the position of carbon number 5 and position of carbon number 3, there is a phosphate. There is a phosphate. So that bond is what we call phosphodiester bond. Phosphodiester bond. Phosphodiester bond. So mind you, as you can see from this strand is oriented from 5' to 3', but this is oriented from down to upward. They are antiparallel. But the direction of growth is from 5' to 3'. Do you know the reason? Do you know the reason why the direction of synthesis is from 5 to 3 direction? Okay, so it is the direction of synthesis. Always new nucleotide will be added at the 3 end, because at this position there is free hydroxyl group. Free hydroxyl group is available, so new nucleotide come and attach it at this, then another will come and attach it, another will come and attach it, another will come and attach it. In such a way, it forms a polynucleotide. So the strand is oriented from 5' to 3'. From 5' to 3', they are antiparallel. When one strand is synthesized from 5 direction to this, the other one is from this to this direction. Actually, I will provide this video, this detail in replication process in next lecture. In replication process, I will tell you there is a synthesis, there is a replication process. So new nucleotide will be attached at this position. Because of that reason, it is synthesized from 5 to 3 direction. From 5 to 3 direction. In such a way, it forms what we call a polynucleotide. A polynucleotide. Actually, DNA it is a negative charge. That negative charge is because of phosphate. Its backbone is made up of phosphate sugar. So phosphate is a negative charge. It makes a DNA a negative charge. It makes a DNA a negative charge. This is one nucleotide. So I already told you this. This is a phosphate attached at the position of carbon number 5. New will be attached at this position. So this negative charge is due to a phosphate. There is free hydroxyl group at the 5 end and 3 end. So phosphate attached at this position and at this position. So the strands are double. They wind around each other in antiparallel orientation. For example, if you have the sequence 5' AT GTC 3' on one strand, the opposite strand could have a sequence at this position 3 T A C A G 5. They are antiparallel. Mind you, they are complementary. There is what we call complementarity. Adenine always pair with thymine, guanine always pair with cytosine. They are their pairing is very specific. When I complementarity, that is the pairing. They are very specific. They are very specific. Adenine never pair with guanine. Cytosine never pair with thymine. So always adenine pair with thymine, guanine pair with cytosine. So they form a antiparallel. Okay. As a summary, DNA is a genetic material in any eukaryotic cell. In any eukaryotic cell, it is a genetic material. Mind you, it is a material that stores information from parent to information, that store information and transmit that information from parent to the offspring. So it is a genetic material. It is a double strand. The strand is antiparallel. The nitrogenous bases always found in RNA are adenine, cytosine, guanine, and thymine. The sugar in DNA is deoxyribose. Deoxyribose. Thank you very much watching this video. Like it, subscribe and share it to your friends. Thank you very much.